I'm very excited to introduce Jessica Cline today. She's our research coordinator here at Marine Resources Council. Hailing from Tennessee, she majored in general biology at the University of Tennessee at Chattanooga. While working in environmental protection and restoration was always her goal. The call of oceans and estuaries brought her here to Florida, thankfully. She is currently working towards her master's degree in biological oceanography at Florida Institute of Technology. Her expertise is benthic ecology, and her research focuses on using benthic and sessile organisms as indicators of coastal restoration success. And she will tell us all what that means. So I'm going to start with Q&A, and we'll give this over to Jessica. Thank you.
Thank you, Mara. So welcome in everybody in the house. Thank you for those of us that are tuning in on Zoom. Like she said, I am Jess, and today we're going to talk about benthic infaunal responses to shoreline restoration. If you do not know what benthic infaunal are, don't worry, we're going to get into it. Can you click it again? Yep. There we go. So this slide's a little superfluous now that we've gotten this nice intro, but I am the research coordinator here at MRC. I've been here as of this month 2 years, so I've been working in the lagoon for a while now. My job basically is just to manage the research projects we do here on site at the Lagoon House and out in the lagoon itself. Pretty much all the data we collect goes through me. I also coordinate our Lagoon Watch program, so our volunteer monitors that collect water quality data. I run that program now as well.
Oh, sorry, you were there. Got it.
There we go. So we mentioned all this before. My degree is in general biology. I also have a minor in environmental science, and I moved here in 2022. So I've been around for a while, and my job is super cool because I get to do out in the field stuff and kind of the fun sharing data kind of aspects of science. So today we're going to talk about the restoration site that we have here at the Lagoon House here in Palm Bay. We are practicing what we preach. We have restored our shoreline here on this property. We're going to talk about benthic infauna, what they are, and how we use them as bioindicators to kind of see how well our restoration is doing. So we don't want to hurt the animals in the area. We want to make life better for them and protect our shoreline. And lastly, we're going to talk about some data from this location. It's been in place for about a year, so we have a nice look at how well it's doing, and we're going to talk through that today. So where is it? If you walked in today, you probably didn't see it. It is on the shoreline behind the woods from here. So there's actually an ice shell midden in these woods. It's a St. John's property. It's a bluff cliff there. So it's not a nice shoreline sloping down. It's about a 30-foot drop to get from where the land is down to the water. And we noticed it was eroding, and we chose to restore this site because we want to protect that midden that's up there from collapsing into the lagoon, since it is a federally protected archaeological site. And for those of us that are looking, this is US 1. So we're right here on the edge of the lagoon. So this is what it looked like standing in the site before we did anything to it. When I took these pictures, I was standing just in the middle of the site. So the picture on the left is facing to the south, and the picture on the right is facing up to the north. You can actually see the 192 causeway up there. So you'll notice that most of the beach here is bare sand. It doesn't have any plants on it. There is nothing holding it there. So actually between when I took these pictures and when we restored this area, we had a lot more of our beach get eroded. Actually up to this forked palm tree, I'm going to use this as a landmark through all the pictures you see. It actually eroded up into here before we were able to get in there and restore it. We want our shoreline to be covered in plants like mangroves. So further down in our terrace shoreline, those mangroves come all the way out to the water line. They're holding the sediment there. No erosion, it's doing great. For those of us here, the Lagoon House is actually kind of up here in these pictures. So what did we do? We restored it, we've already mentioned that. It took a couple months to permit it. Since we were putting in not just plants, we put in human-made artificial structures, we did have to go through permitting. So we are permitted with the US Army Corps and with the Florida Department of Environmental Protection. So this site is registered with both of those departments. We also put in the breakwaters. You see them here in this picture. We're going to talk a lot more about them in just a second. That was in March of last year. So a little over a year ago. And then we planted mangroves in July of last year. You see them here in this photo as well. So the reef arches, we have a breakwater here in this area, not a seawall. So what's important about that is a breakwater is not stopping the wave energy. It is simply slowing it down. We don't want to disconnect our habitat, and this allows water to move through back and forth almost the way it normally would. But because the wave energy is being slowed down, that allows sediment accretion to happen, so beach growth. When water has a lot of energy in it, it can carry sand with it. And as that energy slows down, as it hits our reef arches, that sand can then drop out of the water column behind it and build our beach. They are composed of CSA concrete, so that is marine safe, pH neutral, so we get all kinds of animals that want to settle on them, and they will become a living reef in and of themselves. We have 37 of these units and 3 50-foot sections, so there's 50 feet of them, a 10-foot break, 50 feet, 10-foot break, and then the last one. They're not super visible yet, but there are the 3 sections there. They are 4 feet wide and 6 feet long, and about 2.5 feet tall, and they weigh 1,200 pounds each. We are really not worried about tropical storms coming in here and pulling these out. They're all hooked together. They're not going anywhere. And then, of course, we planted mangroves as well in last July. That was sponsored by Ideas for Us out of Orlando, so thank you to them. We had over 30 volunteers that day, and they planted 130 red mangroves in 2 hours. And that did include them literally ferrying them up the shoreline from the south all the way to the north. So they did it very fast, and they did an excellent job. And then we had all of our mangroves in our site. So there's that same picture from before zoomed in. All the pieces are in place, but this is an experiment. This is my master's thesis. So not all of the sections are identical. We have different treatment groups of different types of restoration. So this is just one section of it. We're going to flip over to a conceptual map of what it looks like, kind of if you're looking at it from the sky. So that picture is of this area right here. But because there are different groups in our experimental design, there are some areas that are planted with just red mangroves. That's what we would call a living shoreline. We didn't have any kind of human-made infrastructure added there. That is the plants by themselves. There is also one section in the northern end that only has the reef arches that we did not plant mangroves behind. And then there are 2 sections in the middle that are hybrid living shorelines. So that is the marriage of our engineered product and our natural mangroves working together to keep the shoreline there and enhance the habitat. And like any good experiment, there is a blank site, a control site. That is, we left it totally untouched so we could have a comparison later to what would happen if we didn't do anything. So we have to collect data on this shoreline so that I can actually write my thesis. And how we do that is every month I collect water quality and sediment pen data. So we're going to take water quality with this. It is a multi-parameter water quality checker. Very fancy word. It means this end goes in the water. It's got all the probes on it. It's down here. And then this end reads off all the information. That's going to be things like temperature, salinity, dissolved oxygen, pH, turbidity, all those classic things we want to know about the water, just as we're going along through our experiment. We also measure sediment pins. So that's this piece of PVC you see right here in the photo with me. That is a method from the US Geological Survey. And what that is, it is a 6-foot pole of PVC that we put into the ground. So 36 inches of it stuck out. So every time I go measure that, I literally take a tape measure, measure from the top of the pin, not to the surface of the water, but to the surface of the sand down underneath the water. So if that distance gets longer each time I measure it, we're losing sand. It's eroding out from the area. If that number is shorter, then we're gaining sand. The beach is growing. Very simple way to do that, and it gives us very, very clear data. So we do this monthly, and then every quarter, so every 3 months of the year, we go out and collect my sediment samples. Not going to talk about that a lot today, but I just collect that with a scoop. I take those samples over to FIT, and I look at the water content of the sediment and the organic matter. So that's a hot button topic around here with muck. We don't want to see muck. That's actually the exact opposite of the issue that was happening in this site, is it had no organic matter when we started. So that's something I'm monitoring as well. And then we collect our infauna. And we do that with this. It is called a petite ponar grabber. It is essentially like a claw machine for sand. So we kind of toss it down on the ground, pull up on the rope, and its little arms will actually close and pull up the sand with it. And then we're going to sieve it in the field, kind of like panning for gold in a sieve. So it lets some of the sand out, but I get to keep the animals that I want to look at in there for processing later. And then kind of informally, just because we want to keep an eye on it, we also survey the epifauna, so the things that are growing on the reef arches as they become a living reef. So we get a lot of algae, like you see here, that likes to accumulate on it. They also tend to collect mangrove propagules. So we're hoping one day maybe the forest will kind of emerge out of the reef arches, and that would be super cool. We also see all our classic settling animals like barnacles, mussels, oysters. When I took this photo, our oysters were only about the size of a quarter. Our oysters that are down there now are about 3 inches long. So they've done extremely well. They can seed this product for oysters. We did not need to do that because we actually had a log just kind of in the area that the spat came from there and settled on our reef arches, and they're doing super well. We also get tons of little critters like snails, amphipods, isopods. They like to crawl all over them because it's a nice space for them to be. So I've tossed this word around a couple times. What are benthic infauna? Show of hands, does anybody know? Okay, that's my absolute favorite response because I get to be the one to introduce you guys to these. So if you're walking on the lagoon shoreline and you're walking on wet sand, there's a whole other world of critters that are living beneath your feet. And they are super duper cool because I studied them and that's my entire life. Yep. So there are a couple major groups that we're going to discuss throughout the day. First one is going to be polychaetes. So these are segmented worms. So this one up here in the top left is one, and down here in the bottom right as well. This one has actually built a tube out of pieces of sand. You can see his mouth part's kind of there. There are also going to be things like arthropods that include amphipods like this, humations, mysid shrimp, and even this is actually it's not a shell really, it's a carapace. This is an ostropod, so bean clam. So it has a little kind of insect-like looking animal that's in there. And then we also have mollusks, so things like snails, like this one, and our classic bivalves, those two-shelled animals that we're familiar with. We see them on the beach all the time. But these are really, really, really tiny. Benthic infauna are super important. They provide a lot of valuable ecosystem functions. They're going to help cycle nutrients through the environment. They're going to bioturbate our sediments, so they're going to dig around and make room not only for themselves, but other animals. They're going to help bring oxygen down into the sediments as well. And most importantly, they tend to be the very base of our food chain. So if we have no infauna, we're not going to have those fish that feed on them, and then the bigger fish, sharks, dolphins, birds, whatever else on top of that. But uniquely enough, we can use them as bioindicators. So that just means it's an animal, a community of animals that we can use to kind of gauge how healthy the environment is based on what animals are there or not there. So if we're looking at an unhealthy environment, we're going to see our infauna community is dominated by worms. So that's going to be those polychaetes from before, or roundworms like nematodes that are the absolute bane of my existence because they're super hard to see. These animals tend to be really, really pollution tolerant, whether that's that organic matter I talked about just a second ago, or if that's chemical runoff from fertilizers, plants, anything like that. They also tend to be detritivores, so they're going to be scavengers. They're going to be eating just the dead organic material that's there. We're not getting that kind of trophic diversity. We don't have herbivores that eat plants. We don't have the carnivores. So they're kind of lacking diversity just like that when the worms are by themselves. And they also don't need a lot of oxygen. So other animals that we want to see in the area need oxygen. These guys don't need quite so much. But that's not to say these animals are bad. They're actually super cool. They're not parasitic. They're not invasive. We just know that when we see them by themselves, the environment's lacking something and we probably need to head into that area to do some restoration. Compare that to healthy environments, we tend to see higher species diversity. We tend to see more things like the arthropods and mollusks pictured over here come into that environment, and there will be less worms in the community. These animals tend to not be as pollution tolerant. They want clearer waters to help them respire. They need that higher dissolved oxygen, just like fish and crabs and all that. And they want more stable sediments. So they want to live down in the sand, and if that sand is moving back and forth, they don't want to live there, just like we wouldn't want to build a house on shifting sands. So they want their home to be stable too, and if our beach is constantly eroding, they don't want to live there. But all of the taxa, including worms, need to be there to perform all those ecosystem services. So that's how we know that these healthy environments are doing what they need to do, because they have this nice mix of all the animals that should be there. So I have all my data, I've done my field work, we've collected all of our samples and brought them back up here to the Lagoon House. How do we analyze it? Well, the first step is I get to spend absolutely hours sitting at the scope in the back room, counting through all of the samples and digging out the infauna. So this is a stereo microscope, so it's not super duper zoomed in like a normal microscope. It's a little bit further back, it doesn't magnify quite as far. But the nifty thing about this is it has a camera attachment on it. So that's how we get all those super cool pictures that I was showing you before. And then I can also see what I'm doing on the screen if I don't want to look through the eyepieces. So I count and record all the species that I find there, and that ends up becoming my community data matrix that I'm going to run codes on later. But to do that, I have to combine all of my water quality data, all of my sediment data, and all of the infauna. And that ends up kind of looking like nightmare fuel when you do it in a computer. So we do have to do it by coding, doing statistical testing with hypothesis. We're not going to talk about this because we'd be here absolutely all afternoon. We're going to skip to the results of that. So the best way for us to talk about infauna is to talk about a year-to-year basis. So just like birds that lay their eggs only in the spring, Infaunal have a reproductive bloom as well. So they tend to have the highest numbers in spring and summer. So I don't want to compare a set of samples from May to a set of samples from December because they're going to automatically be different. So when we're looking at Infaunal today, we're mostly going to be looking at it from last February, before we did any restoration, to this February, about a year later. So we go through. But we're going to kick it back to simply beach growth because that's the easiest thing to see the change. So these are some drone images provided by Reef Arches, actually while they were still installing them. That last breakwater ad's not even fully complete yet. I'm going to draw your attention to these two red dots that are up here on the shoreline. That is that forked palm tree we saw before. This is another log, but we're going to use them as landmarks. So this is what it looked like as they were deploying it in March. And about 4 months later, we had all this filled in sand. So shoreline came from back here all the way up against the breakwater, filled in here and here next to those dots. And it's a little hard to see, but it filled in back here as well. And this is the area that we're continuing to grow even now. So kind of amazing horizontal growth out. We did not bring in field trucks. This was just where that wave energy was carrying sand in. It slowed down as I hit our breakwater and all of that sand dropped out behind our area. Looking at that from the ground, that is day 0. So before they put in any of the reef arches, again, that nice palm tree is going to help us see where we are. 45 days later, it looked like that. Nice, you can actually walk on it and not be in the water. So there's that same branch that's down on the ground. We did go back and remove the tire, don't worry. That side is clean now. And then 100 days later, it was this very wide 10 to 15 foot beach down there that was filled in. And we're actually, probably a little harder to see, but we're actually getting grasses coming down off of the bluff because it was stable enough for them to root down there. So very, very cool. And that only took 100 days in our wave climate. We want to look at that. So that was horizontal beach growth. We were looking just the distance that it grew out. It also grew vertically because it came out of the water, right? So before we did any restoration, before the breakwaters, before the mangroves, anything like that, we had those pins out there. So I was taking those measurements. So I had them out there for about a month before I took this. And 2 of our areas were experiencing up to 2 inches of erosion in a month. So not great. Compare that to another one from this next graph is going to be from October of 2024. Anything above 0 is beach growth from where it started. And this is actually 1 week after Hurricane Milton went through. So before Hurricane Milton, we were kind of trending all around 4 inches of growth. And actually the winds forced so much sediment up behind our reef arches and they got caught there and it stayed. So as we're looking at these sites 1 and 2, as we go forward, we're going to be right just where the mangroves are by themselves. Site 3 is that negative control. Sites 4 and 5 are the hybrid that have the reef arches and the mangroves in them. And site 6 is where the breakwater is by itself. You'll notice site 3 also grew in some sediment. So the reef arches breakwaters do not scour off of the sides. They actually continue to build beach down to either side, which is really excellent when we're talking about shoreline restoration. So site 3 is getting some positive effects from both of the areas on the sides of it. And we'll see that with the Infaunal as well. A lot of beach growth. So our community looked like this last February. Remember I said worm dominated was bad, not healthy environment. So the red and the yellow are worms. Those are nematodes. So these roundworms and polychaetes, those segmented worms. I counted 27,000 nematodes that quarter. Not the most fun thing to do, but really important to note here is the arthropods and mollusks, those kind of more sensitive animals, only took up about 1% of the community each. So 98% of that is going to be those worms. So we knew looking at this, there was something that needed to be done here. We needed to stabilize our sediments, get in some trees to really kind of beef this area back up so the Infaunal would want to live there. I'm going to draw your attention real fast to this others category. That's going to be things like foraminifera, tunicates, those animals that tend to be rarer anyway. And they're actually so rare in this site when we started that unless you've got better eyesight than me, you can't see the orange wedge on this graph because there were only 32 of them that were counted that quarter. So we're going to stay season to season. So instead of going into May, we're going to flip to next February. It looks a little different, a little more spread out. So it is still worm dominated. We've still got our nematodes and our polychaetes taking up the largest section, but not quite as much as before. And polychaetes tend to displace some of the nematodes, so that's great. When we're looking at this graph, nematodes is one species of nematode versus polychaetes, there's several different species in there. So it is more diverse, just as this section has expanded. The mollusks are still hanging out about 1% of the community, so they didn't change a whole lot yet. There are more of those other taxa. So there's the tiniest orange sliver there at the edge of the red. We jumped up from 32 organisms to 162. But the biggest part of this is the arthropods jumped up to 9% of the community. That nice blue wedge was really, really great for us to see. Okay, so we've seen our community composition kind of as a pie graph. Now we're gonna break it down by the types of restoration. So these are those site numbers we saw before. So we're gonna look at the Shannon Diversity Index value for these sites. And that is just a number based on the number of species that are in the site and the number of individuals that we counted, so their abundance. It's not taking into account what types of animals it is. It's strictly a numbers-based calculation there. So when we're looking at this graph and the next one, the color is gonna denote the restoration type. So whether that's living shoreline, the breakwater alone, or the hybrid living shoreline. You'll notice all of the ones on this graph are gray because this was before anything was done. So when we're looking at this scale, most typical ecosystems range on this scale from about 1.5 to 3.5. Brownie face, that is the bottom threshold, 1.5. So we knew right off the bat that this ecosystem needed some help just to even get it up to that functional diversity where we're getting all of those ecosystem services that our Infaunal provide. A year later, there are a lot taller bars here on my graph and they're more colored because we actually did restoration, hooray. So we increased our diversity values across all those treatment groups. Like I mentioned before, the control does get some positive effects from the things on either side of it, so it actually increased in diversity as well. And one site now meets that very minimum threshold of 1.5 on the Shannon scale, and it is site 5. So that is a hybrid site. That is the most intense form of restoration that we did with the breakwaters and the mangroves there together. Again, this has only been in place for a year. To see this much of a change has been absolutely amazing. These numbers will continue to go as that area is still stable, more things can move in and colonize. And the next two groups to probably cross that threshold are going to probably be those other two sites with the breakwaters where the sediment is more stable. So sites 4 and 6 are the next ones that are close to that category. But all the sites are more diverse. But does that mean the community is the same? No, it does not. The type of restoration we did, so those different treatment groups, actually affects which animals live there, which is absolutely fascinating. And we're going to dive into what this graph is, because right now it kind of just looks like a bunch of dots scattered around. This is the result of a Permanova test. So that crazy nightmare computer loadout I showed you, that's what this does. So it is a permutational multivariate analysis of variance. All that means, you clicked off of it.
Oh, sorry. Sorry, got it.
It's okay. All that means is it lets us look for differences between different groups of data. And in this case, the groups of data we're looking at are the communities of Infaunal between all of those treatment groups. It assigns me a number to all of the different factors that might affect the Infaunal community. So maybe the water temperature, or how much the sediment has moved, or in this case, the deployment type, so the type of restoration. That number is called the p-value. And we want that number when we're testing to be less than 0.05. That means there's a less than 5% chance that the results we're getting are due to random error. Or on the flip side, the model is 95% confident that that's why this change is occurring. So, drum roll. Wait. Okay. The p-value for this was 0.01, so a lot lower than 0.05, which is great. So that means our deployment type, the type of restoration, was having an effect on which organisms lived where, which is super fascinating. So as we're looking at this graph, the red dots are going to be that blank control site. So the area that we did no restoration to. The green ones are going to be where the breakwaters by itself. The blue dots are the living shoreline with just the mangroves. And the purple dots are where the mangroves and the breakwater are together in the hybrid shoreline. So as we're looking at this, excuse me, closer clusters of dots on the graph mean that the communities are more similar to each other. So if we look first at the red and the green, so the breakwater and the blank site, those communities are super close together. They're all right there on the right side of the graph. So we know the same types of animals like to live where there's either no restoration or a breakwater. Compare that to the blue and the purple, that are the mangroves and the hybrid site. They are way more spread out. They take up this entire plot. So we know not only are those two communities different from each other, they're also different from that original breakwater blank only area. So very interesting. So as I've been looking at this, I sit and count, the Infaunal under the scope for hours at a time, right? I've noticed that I tend to see more of those arthropods hanging out down here just where the mangroves are. I also tend to see more of the mollusks, the things with soft bodies and shells hanging out by the breakwaters. just our community has kind of, it's all increased in diversity, but they've kind of separated themselves out by group depending on what's there, which is really kind of fascinating. Neither one of these things is bad, they're just different from each other. And that's super cool as a scientist to try and get to figure out why that is. At this point in time, my best guess is that these animals need more stable sediment. And because those are where the breakwaters are, that is where it's going to be the most stable. That's the more intense form of restoration. Our shoreline is more anchored in there. And that's why they like that space. There's also a difference in abundance, so just the number of organisms that I counted there. And for this, we're only going to look at the mangroves and the hybrids, because those were 2 sites each, so I don't want to mess with our counts here by including sites that had only 1 set of samples from it. So in the mangrove sites, I counted just over 8,000 animals, which is still a lot than a year ago. But compared to the almost 13,000 that I counted that lived up where the breakwaters are, you know, there's a clear difference there. And again, my hypothesis at this point in time is that this is providing a more stable home for those animals. There's more space for them to get in there and colonize and live their lives peacefully and not be eroded away with the beach. So it's working. The diversity is increased across all the groups. Why is it working? If you're new to MRC, this is going to be new information to you. You've been around for a while. You've probably heard all of this before. So we love mangroves here at MRC. They are going to stabilize our shorelines. They're going to do that by reducing erosion, because at least the red mangroves have those big walking prop roots. They're going to use to hold the sand in place, keep it from washing out. That's also going to help you a lot with flood protection during storms, hurricanes, all that kind of stuff. They are also going to filter water as it enters the lagoon. Those roots are acting like a big sieve, so as our water comes into the lagoon from the land, it's going to trap sediments and pollutants right there instead of letting them go into the lagoon where it can harm our fish and dolphins and all that. They are also habitat spaces for most of the animals that we think about, fish, crabs. Actually, most of our commercial fish spend at least part of their lifetime in the mangroves when they're young, because those roots, they can hide in there from predators. They can grow up and then move out of there. And lastly, they are absolute masters at carbon sequestration. So that just means that they are really, really, really good at taking carbon dioxide from the air. When they do photosynthesis, they bring it in, and instead of just releasing it back out, they put it back into the ground through their roots. And if the carbon is in the ground and not in the air, it's not affecting our climate, it's not making our storms any worse. So we want it to be down in the ground. And mangrove ecosystems are the poster child for carbon sequestration. Moving on to breakwaters, they do the same thing, but a little differently. So they're going to stabilize the shoreline as well, and they're really going to allow that beach growth behind them, like we were seeing. Mangroves can do it, but it won't be quite as rapid. And they're also going to minimize scouring. So compared to a seawall or a abutment, they're not going to undercut from the front, along the front side of it. And like we saw that beach growth to the sides, they're not going to cut out neighboring properties on the sides, which is absolutely excellent. They are also going to provide habitat space, but for a little bit different animals there. We do see fish that like to hang around them because it is a protected space for them to go in and out of the reef arches. But also that settlement space for sessile organisms, those things that will root down and stay there their whole life. So mussels, barnacles, especially our oysters, anything that's going to filter our water and make it cleaner, we want to settle out on our breakwaters. And lastly, most importantly, breakwaters do not cause habitat fragmentation. So if you put up a seawall, by definition, it is a wall. You're stopping the movement of biological animals and plants from moving where they want to go. With a breakwater, we're just slowing down the wave energy. Everything can still move around it as it normally would. So our area stays biologically active. There's no inhibition of movement or migration through there. And if you do a hybrid living shoreline, you get all of these things. So probably why those sites are the most successful. That site number 5, that one in the middle that actually did meet our threshold, it was the hybrid living shoreline. So it's getting all of this wrapped into one restoration site. So our infauna increased in biodiversity. I know that because I'm looking at the numbers, but I mentioned before, infauna are the base of the food chain, right? So we definitely have seen more fish down here, and with fish come apex predators into the area. So we know we're actually getting this bottom-up effect of ecosystem restoration just by putting in our breakwaters and our mangroves. So our first set of new residents that we're able to see, and we see a lot, are Atlantic stingrays. Do not step on them. They will be very angry with you, but they eat all the things that have come into the area with our reef arches. We also see wading birds. So we've got an egret there and a heron that love to hunt around our reef now because there are fish there for them to eat that were never there before. We also have a resident osprey who comes in, and he gets very angry with me because I'm in his space, and he will swoop down and try to get me. It's really fun. And a lot of people's favorite, since there are fish there for them to hunt.
Oh, sorry, I caught it.
We see dolphins down there all the time now that we didn't used to see before. So this is a pod. You've got 2 adults and a young one, and you can actually see the fish kind of jumping out of their way as they're hunting for them. And that's only about 30 feet off the shoreline. You can see the reef arches there. And they weren't scared of us at all. They were just like, we're here for the food. Oh,
I just started.
yeah, okay. So not only have we seen our food chain increase in its numbers all the way up to our apex predators, we actually, because our sediments were more stable, really a huge thing here in the IRL, so we had a patch of seagrass pop up in front of our reef arches. So this last summer was an ephemeral patch of seagrass, so that means it was only temporary. It was only there from probably about mid-May to August. As our water started to cool back down, we did lose the patch of seagrass, but that was to be expected. This is a patch of Rufia maritima, and it was about the width of my arm span. Nice big patch. That's what it looks like under the water. So you can actually kind of see the fuzzy stuff growing on the blades of the seagrass there. That is their kind of epiphytic community that they like to have. So bacteria and microbes that live on them. And we know when we see all of that together, that patch of seagrass is really happy and healthy because it has all the animals in it that it's supposed to have. And actually, when we sampled the infauna while the patch was there, we found infauna that were specifically associated with seagrass, that people had made that connection before. So our patch was there long enough to attract those seagrass associates to it, which is really, really, really cool. This project, while I will be defending in August, does not end with this. We will continue to monitor it into the future and hopefully watch those diversity values continue to increase. And that's all I have for you guys today. If I don't get a chance to answer your question or you have things you want to tell me later, please reach out to me, Jessica at mrcirl.org, or you can call our office number. And with that, I'll take any questions.
I'm gonna start with a question on Zoom. If you have questions in the audience, if you can hold up your note card, I'll come get it in just a second. But our first question is, can you explain why you did a control in the middle and not on the end?
So, yeah, the way this area was, let me actually flip back a bunch of slides as I'm talking here. So it wasn't ideal. Obviously, it ends up being more of a positive control as it is getting those effects from the sides. But on the very southern end of the area, we had adult mangroves that were already there. So I didn't want that to be interfering with my results. And then actually just the way the land slopes on the other side, there wasn't actually room for me to take samples back there. So we actually had to take samples from kind of in this area, not in the same line as before, because that breakwater had to be at an angle. Super fast. It'd be faster if I did it. Yeah, I don't want to do that. Sorry.
It's going, it's
It's
almost
just very
there.
slow.
This computer's had a lot
Yeah.
of issues today. I'm just happy it's on.
There we go. Okay, so.
Oop,
too
now
far.
you put too many
Now it's too
times.
far. So down here, kind of off to the farthest south side of the area, that's where those adult mangroves are. So if I was taking samples from there, I would have been taking it directly from them, and it wouldn't have been the part that we restored for this project. And then up here, the land actually curves in a way that there weren't samples really in an area that I could take on the other side that would be reliable.
Did you talk about the one on the end?
Yeah.
Yeah, okay, I did. Okay, I have some comments.
Okay.
This is such a great research project. Thank you for the study.
Thank you.
Thank you, Patrick. We love you too. What size and age of mangroves were planted?
That's a great question. So we planted mostly 3-gallon red mangroves. So they were probably in the realm of 3 years old, 3 to 5 years old. They were about 3 feet tall. And we tried to selectively pick ones that already had at least one prop root so that they would maybe anchor in a little bit faster. The trees that we planted were also treated for the mangrove pathogen diseases that we are experiencing here in Florida now, just to give them the absolute best chance of survival in this area.
Okay, thank you. How much are the breakwater pieces? So one reef arch of the 1,200 pounds.
One reef arch is $2,500.
Yeah, so they have 3 different sizes of this product. The most common is going to be your 1,200 pound, the ones we use here. which are 2,500 a piece at the moment. They also have small ones, which if you're here in person, we have samples of the small ones over there. They're 50 pounds. Those are more for homeowners that want to do it themselves, kind of thing. And then they have an even bigger one that's 7,000 pounds, and that one's actually meant to be sunk, seeded with things like coral and oysters. So our mid-size, the 1,200 pounds, are going to be what most homeowners and municipalities are going to use, and those are currently priced at 2,500 a piece. We do work with that company. We also do the permitting and all sorts of stuff. So if you're interested, please let us know. Ellie, your additional question, will your presentation be available for recording? The answer is yes. After this presentation, it will be uploaded to MRC's YouTube and you can watch it again. And then, are there funds available to homeowners that would like to install breakwaters in front of existing bulkheads? Do you want me to answer that?
Yeah, that one's someone.
So that's kind of my area of expertise today. Unfortunately, there are limited funds available to private homeowners. So there are things like small grants that you can apply for that usually will not cover the entire cost of the project. So let me give you some background on this project. This project was funded with a $50,000 Tourism Development Council grant that the MRC applied for. And those are things that municipalities and other organizations can apply for you too. If you are a private homeowner looking to do this kind of project, I highly recommend you find a local grant writer. They will know the opportunities that are available and set you up in the correct direction. Someone around here is Lee Hale. He's a really great opportunity, or great resource, and I can give you that information if you need it. Let's see. And then, are you seeing differences in dissolved oxygen values by restoration site?
No, it's not varying that much. Our shoreline here, we get a lot of wave action. So whether that's just wind or boat wakes, soil water is really being stirred up and it's got a lot of contact with the atmosphere. So low DO is not tending to be a problem here. It was that no organic matter that was keeping our community kind of repressed before. I've never seen dissolved oxygen anywhere near the lows that would be concerning in this area. Oxygen wise, it's very good.
Yeah, that has to do with the water depth, as well as we have a freshwater stream upland that will bring in oxygen. So this area is not historically hypoxic. It tends to be the deeper areas in the lagoon that get like that. But luckily we don't have those issues.
No, but not.
Does anybody else have presentation specific questions before I answer our last one? Okay, our last question is, what is your annual budget ratio of funding versus donations? And that is a great question, especially in the environment that we're in right now. So MRC is supported by people like you. We are mostly funded by private donations. And we do apply for state and local grants like the SORL money, Tourism Development Council, like this one was funded through, and other small grants. We do not receive federal funding, and we do not receive any benefit from the half-cent sales tax unless we receive a grant from it. So we do not receive that funding directly. We are very lucky here that we have a very sound board of directors. And so they are very responsible with our money, but we are completely funded by donations. So please support us. Any other questions before we let Jessica go? And she'll stick around for a few minutes in person.
I just wondered, do any plans to do another area around here?
Yes, that is a great question. So if you didn't hear that on Zoom, the question was if we're going to continue the restoration on our shoreline. So if you haven't been to Lagoon House before, we have quite a substantial shoreline. Unfortunately, we don't have access to it right now. It was knocked out by Milton. And this is a city property, so we're waiting for the city to reopen that access.
Yeah.
Fred also says great presentation.
Oh, well, this area specifically, actually, after the time of my thesis is done and I'm not collecting data for that anymore, we're hoping actually to put in some breakwater units down here as well to fill in the rest of this area. Probably a different product than this, so we can have a nice side-by-side
In the
comparison.
control area?
Yeah, so
Yeah, we will fill in.
what the goal is, funding dependent,
Yes.
is to fill in the rest of our shoreline. So if you've been here before, we have 3 completed areas. The first is in front of our building here. That was done in the early 2000s. That was our terraced shoreline. And so that exists. And that's currently, and that was in one of her photos where she showed the adult mangroves coming out into the water. So that was installed in 2004. That's about a 200-foot section just to protect that building. Down this way, Hurricane Nicole and Ian really destabilized the bluff, which is why if you've tried to go down our trail recently, you can't. And so the city of Palm Bay is working on getting FEMA funding to restore that area. And then if you go south of that is our Coquina wave break, which we did in 2019. And so we have spaces on either side of the terrace that are still blank. And we'd love to fill it in with other products. We can do side-by-side comparisons of these new products because a lot of them haven't been tested before. So yes, if we get money.
You wanna be the guinea pig.
Yes, we
do want to
want
be the
to.
guinea pig.
That's the research we like to
Yes.
do. We like to do proof of concept. People, it's one thing to hear that one of these shorelines does better than a seawall. It's another thing to have data to back it up.
Yeah.
Yeah,